Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Driving Better Titers and Shorter Timelines with a Robust and Scalable CHO DG44 platform. My name is Sumaya, and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Erigen, who developed the content for this presentation. Erigen Life Sciences is a leading R&D and manufacturing solutions provider for the life sciences industries worldwide. Erigen offers end-to-end integrated or standalone solutions to advance small and large molecule programs from concept to commercialization. Established in 2001, the company operates through a global network of 6 sites in US and India with a team of 3,800 plus employees and 450 plus PhDs. Its expertise and experience have enabled over 450 customers, including 7 of the top 10 pharma companies globally, in advancing their research programs from early discovery through development and commercialization. And now it's my pleasure to introduce our speaker for today's event, and that's Dr. Sufia Karim. Dr. Sufia Karim is Director of Cell Line Development, is Senior Director of Cell Line Development and Late Stage R&D at Erigen. She has more than 18 years experience in the biotech industry, including leading cell line development, cell culture, process development and optimization, and successful tech transfer. Dr. Karim previously worked with Boehringer Ingelheim and Amgen. She earned her MD from Sir Salimullah Medical College, Dhaka, Bangladesh, and her MS in molecular and microbiology from San Jose State University. So now, without further ado, I'd like to pass control to our speaker. So Dr. Karim, whenever you're ready, you can go ahead and get started.
Thank you, Sumaya, for the nice introduction. Good morning, everyone. This is Sufia Karim. So my talk today is on a robust and scalable CHO DG44 platform. Can I have the next slide, please? So as Sumaya mentioned, Erigen is a contract research organization. At Biologics, we start from antibody discovery, optimizations, protein science, analytics, cell line development, process development, all the way to CMC. Our biologics department is situated in Morgan Hill, California, which is occupying in 28,000 square feet building. We have 6 individual CLD suites, 2 upstream processing suites, and a dedicated microbial suite. Other than that, we have our own molecular biology, hybridoma, downstream processing, and analytical lab. And currently, we are in the process of building the GMP facility, which will hold 2 500-liter sub for manufacturing the drug bulk substance. Just to show that we are, as a service company, we are covering from the concept all the way to IND filing. So now, today, I'm going to cover why we are choosing the biologics to develop in the CHO cell line, and what is the role of DG44, how it is coming to our market for biologics. Can I have the next slide, please? So to talk about the DG44, I just wanted to give the outline of my presentation, some history about the CHO, then what are the subclass for CHO, what challenges the DG44 facing, and how we overcome those to come to a robust process, and with all these different kind of CHO, but still why DG44. Next, please. So talking about the history, CHO, as all of you know, it stands for Chinese hamster ovary. It became a hugely popular research tool in the molecular biology community. 65 years from now, in 1957, Theodore Park, who obtained the first a female Chinese hamster from Eleanor Roosevelt Cancer Research Institute at Boston, where he extracted the ovarian cell and start culturing in the petri dish. And he have observed that these cell lines can be easily cultured outside in the petri dish, and it's like dividing nicely, keep it's infinite time he can generate the cell line in outside the hamster. So that makes very interesting in the molecular biology field and very useful for many research purposes. That is the start of a CHO cell line. That was called as a original CHO, that is OriCHO. After that, lot of studies are keep going on how they can be useful in our molecular biology and how in the biotech industry. Then in 1968, Theodore Park and Cow, they developed, clone that original CHO and find a population of the cells that is called CHO-K1, which is lacking a chromosome, which is needed for the glycine synthesis. So that is the origin for the K1 from the original CHO. And then all the works are keep going on and on. So Larry Chassin and Arlab, they in the Columbia University, they actually developed the cell line DUXP11, which they actually did with the chemical mutations of one of the loci, the DHFR gene, which is the dihydrofolate reductase gene, and the other locus was a missense mutation. So that was a very great discovery for the CHO cell line development in the biotech, because when the gene is lacking of the DHFR gene, so if we can introduce one cell line with a exogenous DHFR, that makes a very good selection marker for the rest of the cell. Only the gene of interest with the DHFR, they will survive and the other cells will die. So that was the first CHO cell line which was developed from the CHO-K1 by Larry Chassin and our lab, and start the development of the biologics in CHO cell line. But the problem was there that as the second locus is a missense mutation, the revert back of the DHFR gene simultaneously happened. So the selection became impossible. So then more studies was going on, and in 1983, Larry Chassin, who actually did the gamma radiations and removed both the loci of the DHFR, and the origin of the DG44 starts. So this is the DG44, which is totally, completely lack of the DHFR gene, which is, there is no chance of revert back to the DHFR gene. So the selection is really robust in there. And then only the cells which have the exogenous DHFR gene, they will survive in the media, which is HT negative. That means that glycine, hypoxanthine, and thiamine deficient. So this is the first 1983 DG44 was established and become very popular in the industry. Can you go to the next slide, please? So just to a schematic of CHO cell line, which I was talking in the last slide, that CHO was first developed in 1957, and then the ProCHO was developed, and then CHO-K1 came 1968, and from there, 1980, the DUXP11, which was the one locus deficiency of DHFR, but there was an issue of the spontaneous revert back. At the same time, in the middle portion, you can see that Larry Chassin in 1983, he developed the DHFR, double negative DHFR gene with the DG44. So this is becoming the main cell line to develop all the biologics there. And at the same time, the CHO-K1 cell was also like try to develop like how we can make it more robust, more serum-free adaptation, get the suspension cell adaptation. CHO is also developed. And then all the different kind of study is going on with the CHO cells, how to modify more, how These are the extremely good cell line, which is doable to take all these genetic mutations, and then how much more improvement we can do. And industry is moving all different direction to develop the different sorts of CHO cell line for use of more biologics development. So my focus is mainly towards the middle portion on the DG44, though it was like came very early in 1983, but still it is going on, and still the demand for the DG44. Why is that and how it works? Can I have the next slide, please? So now is CHO. Why CHO is important to us? So if you look at the industry, more than 70% of the recombinant proteins are developed in CHO cell line, whether it is in DUX11, DG44, or different CHO-K1 lineage. But CHO is the main cell line which is more industry is leaning towards. But why CHO? There are more other cells, but why people take the CHO? Couple of the points, like at least 6 points, I must say that it is very easy to culture. They are like both can be cultured in the adherent way as a static culture and also in the serum-free suspension culture. So as they can grow in the suspension culture and in a large scale, in a serum-free chemically defined media, it makes it suitable for manufacturing. So large scale production, not only for the suspension in the large scale, these cell lines are very important that they can handle the variation in the parameter, just like oxygen level, pH level, temperature, or even the high cell density, they can handle it. So that's why it is very suitable for a manufacturing perspective, where we need a tremendous amount of the cell to generate the protein of interest. The next thing is like post-translational modification. This is a very important aspect of the CHO cell line. They have the capability of doing like human-like post-translational modification, which is needed for the proper protein folding, proper glycosylation pattern, which affect the half-life of the cell, of the product, efficacy, and also the stability of the recombinant biologics. The next one is low virus susceptibility. As they're coming from a hamster origin, the risk of propagation of the human virus is very low. And as the virus susceptibility is low, the loss in the production will be is less, and also it increases the safety of the drug. Then the genetic cell engineering. This CHO cell can be easily manipulated genetically to knock down or knockout or post-translational modification to do the recombinant biologics and improve the capability of the cell. And lastly, definitely the history. CHO cell line is one of the most characterized cell line in the industry. Most of the biotherapeutics are already approved from FDA. It's a very good track record of the DG44 and the other CHO cell lines, particularly DG44. More than 50 biotherapeutics are already approved in USA and Europe. So that's why like we are talking about the CHO cell line, all the different lineage of the CHO cell line, but why they are important? Because of their characteristics. Next slide, please. So talking about the biotherapeutics, initially why the biotherapeutics came, because these are all generated in the living cells and their biologics, and initially they started in the E. coli, insect cell, all the murine myeloma cells, sp2, BHK cells, but nowadays, once the CHO is developed, it's moving towards the CHO. In the upper right-hand side, the graph is showing from 1982 to 2004, all the biologics distribution of origin. Like the blue one, you can see the CHO, which is occupying like 30% of the proportion, and E. coli was equivalently important. Most of the earlier stages, people were using E. coli, yeast, miscellaneous mammalian cells, insect cells. But in the lower bottom one, you can see that nowadays, instead of E. coli, yeast, and other cells, CHO cell is capturing most of the market. It is occupying more than 70% of the approved biotherapeutics is in CHO cell. These are more important for the more complex molecule like monoclonal antibodies, cytokines, enzymes, execution, hormones, clotting factor, all can be developed in the CHO cell line because they have the capability of protein folding, glycosylation, like human. Next slide, please. So why DG44? So the pioneer of the DG44, as I mentioned, it is Theodore Park and Larry Chassin, right? So they are easy to grow, they are scalable, they have the FDA approval, but how it works? So as I mentioned, when these cells have no DHFR gene inside, so if the cells are cultured, they need to have the glutamine, hypoxanthine, and thymidine rich media. But once we transfect the exogenous gene with our gene of interest and the DHFR gene along with it, once we transfect bottom, you can see that the cells which has this gene, transfected gene, when we culture without the HT media, the cell didn't have the insert, they all die. So we are selecting the population which will have only the DHFR along with the gene of interest inside. But later on, the selection is not only with the DHFR here. MTX came to the play. MTX is the methotrexate, which is actually suppressing the DHFR activity. Introducing the MTX, the way it works is like the low copy number, which has the gene with the DHFR, they will also die. Only the high copy number of the DHFR with the gene of interest, they will survive. So we are actually using the MTX to amplify the higher copy number genes and make the titer, the expression system, more robust, more robust and more higher titer one. So selection process not only with the DHFR and also with the MTX, we are amplifying the higher gene copy number cells. So that's why DG44 plays a great role in the industry to make the recombinant monoclonal antibody. And it opening the opportunity for revenue or developing the biosimilar opportunity lot. So we can develop the drug with using of the DG44 cell line. Next slide, please. So DG44 has showed you that they are growing, they are developing, but still the titer is low, yield is not that high, and also the complexity of the media. So the media needs additives, a lot of substance, and also when during the cloning, by surviving by one cell to start, it is difficult. So that time they need the serum to grow the cell, and also it takes long time to generate any of the biologics, including the DG44. We have to screen a lot of clones. 2 rounds of cloning takes long duration of developing from the transfection all the way to the clonal cell line. So how we can overcome these challenges? People start working on that on all different aspects. Like how can we improve the media? How can we improve the feed? Feed will help to improve the yield and also the growth pattern, better growth, better yield. Then the other question is thinking about the cell engineering, how to improve the cell line itself, then the vector improvement, and then the process improvement. All these categories start working together to make the cell line more robust, and the challenge we had, the low yield complexity media, that should be recovered. So for the next portion of my talk, I will talk about how we can overcome this challenge, like the growth media or the feed. So a lot of studies were done for the last 5-10 years, and they find all different kind of media, chemically defined media, which is animal component free, and that actually helps boost cell growth and cellular growing in suspension culture with high cell density, and they can be easily scale up to the next level, and the titer also getting better. But still, it takes long timeline because to have a clonal cell line go to the manufacturing, still we are stuck with the 2 rounds of limited dilution, and that takes a long time. So here in the picture, you can see that from one file, we are thawing from the different shaker flasks going up to a whey bag or the sub. It is possible, but still to go to the single cell clone, we are limited with a limited dilution. We need the serum from the beginning at the dilution phase and taking long time. But how we can overcome that? People are still working on that. Go to the next slide, please. So for that, the selection and screening technology improvement has done their cell growth and doubling time. That is also was working, changing the feed, changing the media, how to reach the peak VCT better, how to improve the specific productivity, screen, how can we increase with the better screening technology. Maybe we have to clone the populations. Before cloning, we have to enrich the population, how we can do that. And also definitely the cell engineering, how to improve the actual cell, the whole cell by genetic engineering to have the cell grow better, improve the robustness of the culture, specific productivity can be higher, and also can we modify the glycosylation pattern according to the need and increase the ADCC activity by gene RNAi or the gene deletion of the glucose. So a lot of activities are going on. People are working on all different directions, how to improve this bottleneck for our DG44 platform. Next one, please.
All right, audience members, this will be your first poll question. So you should be seeing that poll question on your screens right now. And that question is, how important is it to assess the quality by early analytics, which may prolong the timeline? And your options are somewhat important, or high priority, or that could be not that important. So whatever your answer may be, go ahead and click on that answer. So once again, audience members, we do have a poll question for you, and that poll question is, how important is it to assess the quality by early analytics, which may prolong the timeline? And your options are somewhat important, or it could be high priority, or not that important. I'll give you a couple more seconds to answer that, audience members, and then we'll go ahead and see the results. Okay, so I see that most of you have voted. I'll go ahead and close this poll now and share the results. And the results are as follows. So 37% of audience members voted for somewhat important, and then 63%, so the majority of audience members voted for high priority, and we had no votes for not that important. So back to you, Dr. Kareem.
Yeah, so very good. I totally agree with the audience. The analytics play a huge role in our biologics. And definitely we have to have our analytics along with when we are developing the cell line. So we can move to the next, how the overcome our challenges in our DG44 media optimization, how they were working. So the goal of the media and feed optimization, how we can improve the cell growth and reach higher peak VCT. The idea is if we can reach higher peak VCT, we will have better titers. And for that, scientists pay real attention on the cell's metabolism, cell's biology, looking at different stages, why the cells need energy at what stage, what material can give the energy for them, how can we shift the metabolic path from growth phase to the production phase. So looking at like the main two cycle, like TCA cycle, pyruvate cycle, lactic acid cycle, all these metabolic pathway people pay attention, and that helps them to develop the media which is rich for the growth, and some media is rich for production. So huge development have happened in the media feed optimization, and now currently many companies are providing very specific for the different cell line, which is good for that particular cell line's growth and also the productivity increasing. Next one. So here is one of the example of the media feed development happen. In the earlier, in the bottom left-hand corner, the slide, you can see the red one, which is showing the growth pattern of the cell. The red one is with the historic media, where you can see that peak VCT reaches about 5 to 6 million, and the culture is lasting 14 days. And then the upper three different blue lines, those are the new optimized media and feed. So the target is to change the peak VCT higher, and that will reflect into the titer. And as you can see, all different three media, the peak VCT ranges now from original was like 6 million, now we range from 8 million, 10 million to 12 million cells. So they reach to the enriched media and feed composition. We are reaching better peak VCT from the original, and on the right hand side on the titer, you can see the red one. Original was 600 mg per liter, and now with the optimized better media feed strategy, it is almost twice the titer, like 1200, 1.2 gram per liter. So definitely media feed with a higher peak VCT, the titer reach, the QP is improved, and that we reach our target. But people are still not satisfied. Can we do much better than that? So studies just keep moving on, and other aspect, a lot of work is going on, how we can improve more and more. So next one, please. So in that respect, there are a lot of things like genome editing, vector engineering, all work is going on other than process or the media. So like genome editing, usually the random integration of the gene inside the CHO cell, DG44 cell. So now people are thinking, can we get more targeted integrations, like how we can put the gene in a specific area. So zinc finger technology came, talent technology came, CRISPR-Cas, you have heard about it, and then the transposon technology. But what are these things? I'm not going to go very detailed, but just to give you some idea about that. So zinc finger technology are made up of the DNA binding domain that recognize and bind to a specific 3 to 4 base pair of the sequence of the DNA. And also, we need a DNA repair gene, which is known as the bacterial end nucleus, FOC1. In the figure on the top left-hand corner, you can see this right, there are 3 small DNA binding proteins connected to the green FOC1 domain. This is the main target for the gene finger nucleus. They call the gene finger nucleus because the 3 domains you can see, they are connected to each other by zinc. So they are the zinc finger technology. So this FOC1 bacterial endonuclease with the DNA binding proteins in fingers, they make a complex and they come to the double-stranded DNA and cut that DNA at the specific site. So basically all this targeted integration is the main thing, is breaking down the double-strand DNA and insert the gene of interest right on that spot. Same thing with the talent technology, which is the transcription activator like effector nuclease. This is exactly, you can see like a zinc finger, but here the DNA binding domains are much larger, not 3-4 protein domain. They have a better, like a longer flanking site of the target. So, and it is coming from a plant protein, plant pathogen. So talent technology also use the same way. They need the FOC1 too, and they cut the DNA, double-stranded DNA of the genome of the CHO cell, and then the gene of interest is inserted into there. Then the same thing is with the meganuclease and the cluster regulatory interspace short intermixing repeats, that is called the CRISPR-Cas9 system. What happened here is, it is the RNA DNA binding rather than the protein DNA binding, which we have seen with the zinc finger or the talent technology. Here, RNA is guided there into the genome, and then they recognize the portion of the defective gene, cut it out, and then also repair the gene and insert the gene when needed. Other than that, all this technology is improving the better integration of the gene to the cell's genome and getting better titer on there. And the vector engineering also play a major role where using of the different promoter, enhancer, introns, better codon optimization technology, using of different elements, that is like PRE, post-translational regulatory elements, or UCOE, that is the ubiquitous chromatin opening elements, that prevents transgene silencing and gives the consistent, stable, high-titer expressions, irrespective of the chromosomal integration site. So all this technology is directing towards the better titer, better integrations, which will improve the titer at the end. Next slide, please. So that goes with the media and the vector or the cell itself. Now there are still the bottlenecks remain in the clone generations. So still it is like a very labor intensive, go through the limited dilution cloning, need serum time, long time. And with that, only few clones are screened. Not too many clones can be screened. Not all the clones are coming from a single clone. So people are working on that, and then the next generation came the clone peaks. So this is an automated machine learning, like a semi-solid media. They pick up the clone by clone, not manually, but through the machine, and it gives a relief that you don't have to manually do a lot of stuff, but at the same time, that didn't give you the 100% clonality proof. So to get the FDA approval, you have to still go through at least one round of limited dilution for the next stage. So to make sure this is coming from a one cell. Then still people still working on that, how we can make it fully serum free and also very proof of tonality makes much easier. The next generation came the cell printer. Cell printer removes the clonings completely. It has the imager with it. So machine is using the microfluiding technology and deposited one cell at a time in all the 96 well. And the condition is getting optimized so that you can screen more clone when it is guaranteed, like all the wells are having starting with a one cell with a picture, then you can have the proof of clonality, doesn't need to be 2 rounds of cloning. So timeline gets shorted. But same thing, like to grow the single cell by itself is very difficult, but more of process optimization, how to make the efficiency more better. We are at this position, we can get 60 to 80% efficiency from one plate. So every plate, if we get 60 or 80 clone, and if we plate like 5 plate, 10 plates, we are screening tons of clone for generating the best population from there for research cell bank making. So recently, with the advancement of the printer, cell printer and imager, that makes our life much easier, make the timeline much shorter. Now it removing the fully one round of cloning, remove 2 months straightly from there. So more single cells are screened. That gives the opportunity to find out the higher producer clone much better. So leading to high titer RCP. Next one, please. So this is just like an case study I am showing you, that with using all the latest technology, better media, better feed, better cloning equipment with cell printer and imager. Now, like our big VCT reach above around 15 million, cells are growing really good. We are screening more clones, and then the titer reach up to 4 grams per liter. So when we started with the 600 mg per liter, then we improved to 2 grams. Now we are up to 4 grams per liter with all the better technology and our clone selection. Next one, please. So now the DG44 platform is improved and established, which can go with a fully chemically defined serum-free media, and it's a robust, scalable platform for the large-scale production. We are reaching a high peak VCT with better specific productivity and ultimately to the higher titer. It shortened the timeline to generate the cell line from transfection to the RCP, and there is no need for any of the serum at all, so fully chemically defined. But still there are competition for the DG44. As I have shown you for the very beginning, other cell lines are coming up, CHO-DS knockout cells are there, CHO-S cells are there, which are also growing much faster. Their peak VCT is reaching higher, but they are our competitor with the DG44. But still, why we are using and why we are talking about DG44. Next one, please.
Thank you, Dr. Karim, and audience members. That brings us to your second poll question that's on your screens now. And that question is, for innovators in DG44, how comfortable are you in pursuing biosimilar using other cell types which might give you better yield? And your options are not likely, or somewhat likely, or that could be very likely. So go ahead and click on the option that suits you best. And once again, that question is, for innovators in DG44, how comfortable are you in pursuing biosimilar using other cell types which might give you better yield? And your options are not likely, or somewhat likely, or very likely. I'll give you a couple more seconds, audience members, to vote on this poll, and then we'll go ahead and see the results. And also giving you a reminder that you can continue sending in questions for Dr. Karim just by using the questions tab in your control panel. So I see that most of you have voted. I'll go ahead and close the poll and share the results. And the results are as follows. So 16% of audience members voted for not likely, and then 63%, so the majority of audience members voted for somewhat likely, and then 21% of audience members voted for very likely. So thank you everyone for participating. Back to you, Dr. Karim.
Yeah, this is not a surprising answer, definitely. I will also agree with you. I will prefer to have if the innovator drug was done in DG44. We will stick to DG44 because the similarity and getting the biosimilar is much easier, much faster than using other cell lines. So now we are going to talk about why DG44 is still in demand, right? Next slide, please. So the biosimilarity opportunity, right? So what is biosimilar? Biosimilars are the drugs which are highly similar to the original biologics in terms of their structure and their function. But why the biologics came to the market, right? So biologics are the treatment of, biologics gives us the treatment which is more targeted because they are designed to interact mostly towards the immune system in a specific way. So the chances that their immunogenicity, their immune reaction or the side effect, those are very less than the other drugs, right? But however, this biologics takes long time to develop, as I've shown you, and when it takes long time, it is very highly expensive for the general population. It takes long time, high cost. How many people can afford it? Just for an example, if I say like for any of the cancer treatment, any of the drugs, per injection cost about $20,000 to $30,000, right? And over like 4 injection, 5 injection, it costs a lot. So it is not available for the all general populations. For the high cost of the biologics, sometimes it is hard for person to get them, and even it might be the best treatment of the disease. To make this drug biologics more affordable to more people, in March 2010, not too early, not too long ago, so Congress passed a law that is called the Biologic Price Competition and Innovation Act, BPCAI, which makes life much easier. FDA makes approval of the drug in a shorter time period, which lowers the cost and affordable for a larger populations. As you know, the demand of the biologics and the biosimilar drugs are increasing, and they're increasing because their potency, their approval in different indications is increasing, and it is generating billions of revenue and opening the opportunities for the biosimilar. In the bottom table, you can see that within last year, so if up to here, we can see the FDA has approved only in the map 463 maps. And the patent is last 5 years, patent expires about 17 molecule, and in the next upcoming years, 13 more molecule of the maps are getting expired. So just before that, all the different companies, all the countries in different continents, they are working to bring more biosimilar for those drugs, which will be more easy to get approval and more available for the market. So is affordable for the whole human being. So that's the reason, like the opportunity for biosimilar and also use of this DG44 cell line, which originally the innovator was developed in, that opens up the opportunity for the DG44 platform. Next slide, please. So here is just a couple of the example of the blockbuster drug which was made in CHO, like adalimumab, Humira was one of the top one, and it was approved long time back. And then etanercept, rituximab, bevacizumab, trastuzumab, epoprotein or debapirotein alpha. These are all like developing the CHO cell line, and their patent is expiring, and some are expired and some are expiring. So these are like blockbuster drug, and this kind of drug gives you the idea like how the biologics change the path, how we can utilize more, get the biosimilar drugs, and be helpful to the community. Next one, please. So to talk about the biosimilarity, as I mentioned, it has to be structurally and functionally similar to the innovator drug. We need analytics. So one of the question was how fast we wanted to do it, right? So for the sake of analytics, development also was going on huge, and which helps us in the development of the biosimilar drugs, which we can start on very early stage to match with the innovator, and a lot of analytics are in the market and available for us. We use the HPLC, CEX, check the glycan profile, mass spectrometry to check the mass, CIEF for chart variant, and top of that, the activity assay. The drug has to be active and potent and similarly working as our innovator's drug. So analytics play a major role in biosimilar, not only in other innovators' drug, but biosimilar, it is extremely important. You need to make sure from the very beginning you are making the drug which is exactly similar to the innovator. It has to be functionally and structurally matched to it. So role of analytics is huge. So just to give you an example, go to the next slide, please. So here is an example of the adalimumab, which was one of the big blockbuster drug. As you can see that the trade name is Humira. On the left-hand corner, you can see that we developed at Aragen adalimumab, where we are using the mass spectrometry to check the mass and comparing with the side-by-side with Humira, which is the original drug. And then with the CIEX, the bottom chromatogram matching with the innovators, same way in glycan profile checking with the helix chromatography or charge variant and size exclusion chromatography using to check everything is checking side by side with the innovator lot. And lastly, on the right-hand side, top and bottom, you can see that binding affinity, binding TNF alpha binding affinity by Forte Bio or the potency assay, which all have to be matched towards the Humira to be saying that our adalimumab is exactly as a biosimilar drug as Humira. So the role of analytics, I cannot say enough that how much important is analytics in the field of biologics, and particularly in the biosimilar. We have to start the analytics at the very early stage when we are at least at the mini pool level pool, not even at the clone. From the pool, we have to populate the best pool before we start cloning to generate the research cell bank. So next slide, please.
Thank you, Dr. Kareem. So audience members, that brings us to our final poll question, and that poll question is on your screens right now. So that question is, how does the new and improved DG44 platform change your perspective? And your options are somewhat, or positively, or remain the same. So once again, how does the new and improved DG44 platform change your perspective? Somewhat, or that could be positively, or remain the same. So go ahead and choose the option that suits you best. Another reminder that you can continue sending in questions for Dr. Kareem. We already received some questions, lots of interesting questions. So if you have more questions, keep sending them in using the questions chat box. So I see that most of you have voted. I'll go ahead and close the poll and share the results. So the results are as follows. 39% of audience members voted for somewhat, 39% also voted positively, and then 22% voted for remain the same. So thank you everyone for participating. Back to you, Dr. Kareem.
Thank you, everyone. Yeah, so as I mentioned, very early, the DG44 was showing like beginning, the titer was low. It was not that robust. That takes time, but with advancement, it is now like 2 to 4-fold increase of the volumetric titer, shortening the timeline. With further improvement, we are reaching there about to GT44 platform at Aragen. Now we are achieving up to 5 grams per liter within 18 weeks of time. So this is something we are launching at our trademark as Raptor 2022, which gives us confidence that CHO DG44 platform is way better than before and way robust and solid and shortening the timeline. But at the end, it is expected that better understanding of the cell biology fueled by advance in the genomic, proteomic, metabolismic, including the application of the gene expression analysis, combined with further improvement in the media, high throughput technology, online monitoring, automation, that all allows the researcher to broaden the experimental design space as well as lower the cost for the biologics development. Thank you very much.
Thank you, Dr. Kareem, for that very insightful presentation. So audience members, that does bring us to our Q&A portion of today's webinar. So if you have more questions, continue using that questions tab and send those questions in. So we've already received some questions. I'll start off with those. And first question, Dr. Kareem, is how high can we push for the titer in DG44?
Very good question. So sky is the limit. As I mentioned, there are a lot of areas we can still work on and people are still working on, particularly like with cell engineering, further media feed optimization, process optimization, as I mentioned, like more automations, all these things will improve the titer. We can go to 10 gram and even more than that, which is not, anything is not limiting the DG44 platform, which we can apply all this technology, cell engineering, genetic vector improvement, media feed improvement, and also the process improvement.
Thank you, Dr. Kareem. And our next question is, have you compared Tigers, or I think that could be titers, between LGG, LGM, and LGA in DG44 system?
Can you please repeat the question?
Sure. Yep. So that question is, have you compared Tigers between LGG, LGM, and LGA in DG44 system?
Yes, we have done a lot of IgG, IgM in the DG44 system. And yeah, depending upon the molecule structure, the titer is varying. But definitely we have established platform for IgM and IgG in DG44.
Fantastic. Thank you once again. And the next question is another interesting question from an audience member. And that question is, what automation have been used to shorten the timeline for clone selection?
So, yeah, very good question. So as I have shown you, the basically clone selection automation is happening during the cloning portion. That is the cell printer using, it is not manual, it is all robotic, microfluidic technology. You can plate one plate within 4 to 5 minutes. It's pretty good. And then all the screening technology, the ELISA, and those are like now getting much easier. Now, instead of ELISA, the octet is coming there in play. It's very fast to do all those things. And the analytics play a huge role in the screening the clone, which a lot of things are high throughput, like, and also in the field of the biospecific, there are a lot of technology. All the analytics can be done in high throughput manner.
Wonderful. And our next question from an audience member, that question is, what advantages does a DHFR selection method have over GSKO, for example?
Very good question. It is like you cannot compare apple to apple here. So DHFR system, one big advantage over the GS is like you have DHFR selection and also the MTX amplification. And this MTX amplification, as I have shown you, that it is populating the high copy number cells that are amplified. So you are getting second round of selection with high copy number, which is absent in the GS selection marker.
Fantastic. And our next question is, how can cell engineering increase the titer?
Very good question. This is the hot topic nowadays. People are moving towards the cell engineering because cell engineering is mainly using here to make the cell more robust, how the metabolism can be changed, what is the technique to improve the product specific productivity per cell. So there are some companies, they are making, like endoplasmic reticulum is the main machinery for generating the protein. So they are engineering the cell line to make the ER portion of the cell is way more. So if you have more ER in your cell, they have more machinery to generate the protein, which will increase the yield. Then other thing, you can increase the like a cell apoptosis, decrease the cell apoptosis. So knock down the gene which is helping in the apoptosis of the cell. So you are making the cell to survive more. When the cells are surviving more, they are staying there longer, they are producing more. So all different way you can use the cell engineering to improve your titer.
Wonderful. Thank you so much for all of those answers, Dr. Kareem. We've reached the end of the Q&A portion of this webinar, but attendees, not to worry. If you have more questions, the team at Aerogen may follow up with you later on. And for further questions, you can use this email address here, so bd@aerogen.com. Go ahead and quickly jot this down. So if you have further questions or more comments, use this email here. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now I'm about to send you a link in your chat box. You'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join us in giving a big thank you again to our speaker today for that fantastic presentation. So thank you, Dr. Sufia Kareem. That was wonderful. Have a great day, everyone.
Thank you, everyone.